male fertility is a topic which pops up in the news every few years. This is getting a bit old now. This is 2017 when a new paper had come out saying that male sperm counts have been dropping over the last 50 years or so, at least in Western countries. And this paper actually sort of moved quite fast. In 2017, this was quite controversial and people weren't really sure whether it's true. Now it seems like it probably is true. But one thing that hasn't changed is that we still don't really know the answer. So I think there's something quite prescient about Newsweek's Who's Killing America's Sperm, and their nice little tagline, and we can't blame it on Trump, or can we? Because it sort of underlines the fact that we really have little idea about male fertility, what might be causing declines in male fertility in people, and then generally sort of underlines the lack of research in this area, at least in people. So there are various reasons why we might want to understand male fertility. One is that there's a bit of an imbalance in lots of fertility-related things. So, for example, when it comes to control over reproduction, so contraception, we've had the pill for women for a long period of time. It had a huge impact on society. But despite every couple of years there being a promise of a new pill that's going to allow male contraception, it still never seems to arise. Similarly, when it comes to assisted reproduction, if couples have some kind of fertility problem, no matter what that problem is, the treatment is always for the woman in vitro fertilization. So even if the man has got low sperm count, it's actually the woman who gets treated. And there's various other applications to controlling fertility. If you can suppress the fertility of things that you don't like insect vectors and pests, or help the fertility of things you do like fluffy endangered species or farm animals, then there's all sorts of potential societal benefits. But the reason that I'm really interested in male fertilization is because I would say that it sits at the front line of natural selection. So if you want to measure the reproductive success of a male, then it doesn't really matter how big its horns are, how good it is at fighting or even attracting females. What it ultimately comes down to is whether the sperm can fertilize the egg. And this gets made much more interesting in lots of species because females mate with multiple males. So this is quite common, quite variable, taxonomically widespread. Might even have been more important in people in the past than we used to think. And what it means is that in lots of species, you get the sperm of multiple males within the female reproductive tract competing for an egg. Now, sperm competition, we know, can shape the evolution of what males transfer to females. So very commonly, it's thought to lead to the evolution of very large numbers of fast-swimming sperm, such as in these red jungle fowl. And it can have really other interesting sperm adaptations, such as in these deer mice, where the sperm form clumps that can swim faster and more powerfully than sperm by themselves and reach the egg more quickly. But sperm don't travel alone. They are often delivered with a cocktail of seminal fluid substances, and these include all sorts of sort of nutritional things like salts and carbs, lipids, and so on. Various bits and pieces made by the males, different types of proteins and peptides. Cells can come across, and even nasty hitchhikers, such as microbes. And these seminal fluid substances can have lots of important effects. So in mammalian species, we know that seminal fluid is important for the sperm. It improves fertility. It's important for the sperm getting into the right storage areas in the female reproductive tract. And when there's sperm competition, it's important for the competitiveness of those sperm. And it can act directly on the female. So it can stimulate ovulation The reason that llamas are here is because they're an example of a species where there are substances in the male seminal fluid that can stimulate females to ovulate. I think it's particularly interesting because llamas are spontaneous ovulators as well. They have a menstrual cycle, but nonetheless, the male seminal fluid can bring on early ovulation. It can affect the shape of female reproductive tracts, has all sorts of immune-related effects, and ultimately influence the fitness of females. And that's just in mammals. If we extend this to invertebrates, particularly insects, then we see that seminal fluid can stimulate egg laying. That's a real common one in lots of insects. It can stimulate females to feed, which is probably associated with egg laying. Reduce their sleep. It can even affect their vision. I think that's from some bee research. Another very common one is it reduces female sexual receptivity, so they're less willing to mate with subsequent males after mating. And it can even stimulate females to be more aggressive. So I'm just going to take a little detour from the main thrust of my talk now to talk a little bit about aggression, because this was something that Ellie Barth, a PhD student in my lab, discovered in the course of her PhD. So here's a little sort of 5-10 minutes on aggression, and I'll come back to talk more about seminal fluid. So both sexes fight in Drosophila and melanogaster, although the I think there's a software update about to happen. A little thing popped up that said something about a software update. Oh dear. I don't have to talk about aggression. It's just get back to the seminal fluid if you like. Or maybe it was the videos that crashed it. Yeah, that's good. Okay, well, if it was the videos that crashed it, we'll find out through this little experiment. Oh, the videos. We can skip to the slide after that. So just imagine that Drosophila fruit flies bite one another. The males and females have got different fighting techniques. The males, they sort of run around waving their wings a bit, and they'll sort of hit one another with their front legs. They're quite fast. The females, they sort of headbutt one another, basically. So they're a little patch of food, and in the middle of that food, there's a blob of live yeast, and the females really want to eat that yeast. And if it's small enough that they can't both eat it at the same time, they'll essentially headbutt one another out of the way. So that's what that little scenario I just described is what aggression looks like in fruit flies. More broadly, female aggression is often associated with reproduction. So pregnant, lactating females, females that have got small offspring, tend to be more aggressive than females at other times of life stage. So to start off with, my former PhD student, now independent researcher, Ellie Barth, asked whether simply having sex increases aggression in female Drosophila. So we took pair of females on a little patch of food with a blob of live yeast in the middle, and then videoed them and counted the number of times that they headbutted one another, or the amount of time that they spent headbutting one another. It turns out that, sure enough, females that are mated are somewhat more aggressive. About 2 or 3 to 3 times, spend about 2 to 3 times more time fighting one another than a pair of females that are mated. And even if only one of them is mated, then aggression is a lot higher. And it turns out that this increased aggression occurs at some point between about 2 and 4 hours after mating. The time series here, and we've got the virgin's yellow, mated at blue. And see, first couple of hours doesn't make any difference. Then at 4 hours, there's suddenly the virgin mated difference, and that persists for at least a week. So one thing that's curious about this particular time point is that this is about the time when females start laying their eggs. So what we thought was maybe it's just the fact that they start laying eggs is like it's mechanistically linked to when they become aggressive. So what we did was use a mutant that can't lay any eggs and tried to see whether aggression was still there after mating. It turns out that these sterile females, OVD females, while overall slightly less aggressive, still show these differences between mated and virgin. So egg production is not an essential requirement for females to be made aggressive after mating. So we thought, well, what else happens at mating? Well, females get sperm and they get seminal fluid proteins. So we looked to see whether it's because of something in the ejaculate. And first of all, we looked to see what happens if females didn't get any sperm, and that showed a pretty convincing effect. If females don't get any sperm, so that females are still mating, they're getting seminal fluid, but they don't get any sperm because the males are sterile, then those females are no more aggressive than our virgins. So it's all about the sperm. But sperm, as well as actually doing their own spermy thing, are also a vehicle for seminal fluid proteins. So one of the proteins that we know that actually sticks to sperm and is then released off the sperm after mating is called sex peptide. So we did another experiment where we kept males that had normal sperm, but they just lacked sex peptide. And what we found was that their aggression rates were this sort of intermediate level here. So sex peptide at least partially explains this boost in aggression after mating. So that's how we came to conclude that amongst all the various other things that seminal fluid proteins do, one of them is stimulating aggression in females. So this is kind of an interesting list of things that, that seminal fluid does. And kind of when you look at it, some of these things sound like, well, from a male perspective, this all sounds good, right? But from a female perspective, some of this sounds a bit odd. So egg laying, that might be a good thing, maybe. But losing sleep, losing vision, that doesn't sound so good. Not sure about sexual receptivity, aggression, it's hard to know. But if you think about this a little bit more, okay, males transfer some stuff to females that reduces female sexual receptivity. From a male perspective, that's great because it helps exclude rivals and delays the onset of sperm competition. So the female doesn't mate, so therefore his sperm don't get displaced or don't encounter competition from rival sperm. From a female perspective, it's not so clear whether this is a good thing or not. Females might accrue benefits from having diversity in the sperm that they've received, or by replacing old or damaged sperm from earlier mating. So maybe the suppression of their receptivity is not such a useful thing. Ovulation and egg laying is a good thing from a male perspective because all they're interested in is the current reproduction of the female. If she mates with other males in the future and his sperm gets displaced, or she just uses up all his sperm, he doesn't really care if seminal fluid reduces her lifespan. But from a female perspective, her lifetime reproductive success depends not just on her current reproduction, but also her future reproduction. So there's a potential conflict between what male's seminal fluid is doing and what female interests are. So there's a potential that it could be a source of conflict between the sexes. And those kind of conflicts can lead to evolutionary arms races. So what I mean by that is that males might evolve a trait, some kind of new seminal fluid protein, for example, and that trait has a harmful effect on female fitness as a side effect of the benefits that it provides the male. So then you'd expect that females should evolve some kind of resistance to that harm, but if that resistance to male harm reduces those benefits to males, then males could be selected to increase the level of that trait, females get harmed more, females evolve more resistance, and then you get this kind of never-ending cycle of increasing harm and increasing resistance. And there's lots of clever mathematicians who've modeled this sort of thing and show the different ways that it can work. So is there actually any evidence that seminal fluid proteins do fit with this kind of scenario? Well, there's some evidence that seminal proteins can harm females. We found evidence that the aforementioned sex peptide, which helps stimulate aggression and various other things in females, can be harmful to females, although subsequent studies have showed different things. So I think the jury is possibly still out on that. But there are several other seminal fluid proteins in Drosophila that are toxic proteins. If you ectopically express them, then they kill the fly. And also in C. elegans, there's evidence of seminal proteins that reduce female lifespan. There's evidence that females can evolve resistance to harm. There's various experimental evolution studies from Holland and Rice and myself, and more recent work which have shown that the patterns of resistance are consistent with resistance to harmful seminal fluid in the changes to female gene expression. And we definitely know that seminal proteins evolve really rapidly. They evolve really, really rapidly, not just in fruit flies, but across insects and in other sexually reproducing species. So that's why I think seminal fluid proteins are quite interesting. We know that they've got potent effects, but a lot of what we know about their effects on females come from what I'm calling here removal studies. So either you sort of knock them out or knock them down, or chop out a gland that produces them or something like that. So about 15 years ago, I suppose now, colleagues and myself started wondering about what about natural situations where you don't tend to have a binary presence or absence of proteins. You have either alleelic variance or you have quantitative variation. We started thinking about the quantitative variation. So does the actual quantity of seminal proteins, total amount that males are transferring, or the composition, so the relative abundances of different proteins in the cocktail mix, does that actually matter? One reason we were thinking about this was because males have got a lot of potential for adjusting that mix. So in various different species, males produce seminal fluid from a whole range of different glands. I forget quite how many there are here, 1, 2, 3, 4, 5 different glands in humans, and a surprisingly similar number in the fly. These are the accessory glands, the ones that produce most of the seminal proteins. But within that, there's 2 different cell types that have quite different functions. And then there's some proteins are produced in the ejaculatory duct down here. There's a thing at the bottom called the ejaculatory bulb that produces other ones. And some come from the testes as well. So the males could potentially alter what they're transferring to females by adjusting what comes from all these different places. And we also don't really know why males make them in all these different places. So we started looking into this problem. And to start off with, we just used a couple of proteins that we had antibodies for, sex peptide again, and another one called ovulin. And we showed that, fair enough, these things do vary in quantity in different scenarios. Males can transfer different amounts, and that can sometimes lead to different responses. So that was a little while ago, and that was fine for the time. But then we started thinking, well, 2 is a rather feeble number of proteins to be looking at. So we decided to get into the murky world of proteomics. And what we did was develop a method to look at the seminal proteome, which has various pros and cons. So this is what we did. This is a male. This is a little cartoon of his accessory glands and ejaculatory duct that produce the majority of the seminal proteins. And we either kept males unmated, and then we analyzed the glands from these virgin males, or we allowed those some like matched males a experimental mating where they transfer some of that fluid over to females, and then immediately after mating we flash froze those males, and then analyzed those samples and subjected them to quantitative proteomic analysis. So what we're trying to do here is get a measure of 2 things. One is what's the quantity and composition of the proteins that males are producing in the first place, and what is the quantity and composition of what they're transferring to females by comparing the difference between these virgin and mated glands. So this is sort of slightly indirect because we're not actually taking a sample from the female. But the advantage of doing it this way is that you get lots of protein because there's a lot more protein in the male than there is in the female. And also that in the female, lots of proteins get rather rapidly degraded and chopped up into small bits. And that might alter your ability to detect those proteins, or they get transported to different places. So there are pros and cons with this approach, but we thought we'd give it a bash. And here's, well, one thing that we thought we would do, just to start off with, was check that we can detect seminal proteins using this method. What we actually did was find a whole bunch of new seminal proteins, which was quite fun. So this is a volcano plot of from one of our sample runs. These colorful dots on the right-hand side are proteins that are more abundant in the virgin males than in the mated males. In other words, the ones that there's evidence that they're losing stuff when they mate. And rather happily, most of the seminal proteins we already know about that we could detect fall into this cluster here. But you can also see there's a whole bunch of other proteins up there. And the ones that we've colored in red are ones that, from this data set, some other ones, and a bit of bioinformatics, we concluded were pretty good candidates for new seminal proteins. And not only that, when you actually sort of lay out the abundance of all these proteins, the ones that we already knew about tended to be the ones that were very highly abundant, but the ones that we were discovering anew tended to be fairly low abundance ones. So that kind of suggests that this approach is quite a sensitive one, because we're detecting proteins that hadn't previously been identified from various other proteomic analyses. So that was nice. We found some new seminal proteins, but what we actually did this for was so that we could use this approach to address some interesting evolutionary or behavioral questions. And so I'm just going to give you one example of that now. We've done a few studies using this approach, but you know, we've all got homes to go to tonight. So I'm going to focus on this one, the problem of how should males respond to varying levels of competition for fertilization. This is quite an old question that we're sort of revisiting with our new approach to see what we could find. So the problem from a male perspective is that you might be by yourself, you might have a single rival around, or you might have loads of rivals around. And this makes a difference when you come to mate with a female, because it affects the risk that you're going to encounter sperm competition from rivals. And there's some sort of long-standing and fairly well-established theory about what males should do in these different scenarios. This theory pertains to sperm rather than seminal fluid, but I'll just explain what the theory about sperm is. The idea is that if there So this is the total number of males present. So 1 means that you've got no rivals, 2 means that you've got a single rival, and so on. So if there's no rivals around, it's just you, then you can theoretically transfer a rather small number of sperm. You just need to transfer enough to fertilize a female's eggs. A male in that scenario can sort of conserve his resources. If there's a single rival around, a male should really maximize the number of sperm he can transfer to the female, because under that scenario, it's basically a 50-50 raffle, all else being equal. So if you got the most tickets out of that raffle, then you win most of the fertilizations. However, as the number of males increases, then there are diminishing returns. Males can't simply overwhelm the number of sperm from 9 or 10 rivals. So under that scenario, then males should actually decrease the number of sperm that they transfer. So you have this kind of shape of not very much, lots, and then a little bit of a decrease. There we go. So I'm calling this no competition, where there's a male's by himself, low competition, where there's a single rival, and then high competition, where there are lots of rivals. And we did this in Drosophila and Melanogaster, and just to show you that, hopefully this won't crash the presentation, show you that sperm competition happens in this species. This is a really nice video from Molly Marnier's 2010 science paper. It shows the sperm moving around a bit of a female reproductive tract, and this female had mated to 2 different males, one that produces green fluorescent sperm, another that produces red fluorescent sperm. And you can see them, both those sets of sperm, swimming around in the same bit of female reproductive tract. The red are a little bit harder to see, but they're there. So in this species, sperm of 2 different males actually go into the same sperm storage organ, they mix together, and then the female is using the sperm from both these males to fertilize her eggs. So what we did was simply expose males to different competition scenarios. Either they were by themselves, they had a single rival, or they had 7 rivals. So it's no competition, low competition, high competition. We kept them like that for a few days and then saw what they transferred to females. We counted the number of sperm, and we used our proteomic approach to look at the seminal fluid. So in case you ever wondered what sperm transferred to female looks like, it's like this. These are all the sperm heads labeled with green fluorescent protein. This is the bursa, like the main area that the sperm gets transferred to. So it's just after mating. And then over here, this is one of the sperm storage organs. So a few of the sperm have swum over here and are already in storage. And if this female hadn't been flash frozen, then a lot more of these sperm would have swum over, gone over there. So my PhD student at the time, Ben Hopkins, he actually counted all these by hand. There's a few thousand in there. I mean, he had like some nice confocal images to do it from, but couldn't work out an automated way, sadly. And this is what the data look like. So when males experience competition, they boost the number of sperm that they transfer to females. And then if you sort of squint a bit and you're feeling a bit generous, maybe it decreases slightly when there's high levels of competition. So this is at least not inconsistent with the theory. Obviously, the effect sizes are way, way, way smaller than the maths suggests, but it's sort of broadly in the same kind of area. So that was kind of interesting in itself, but what we really cared about is what the seminal fluid is doing. And what we found was that it's a different pattern. So rather than maximizing, let's go back to the sperm. Here we found that males were maximizing sperm under low competition. We found that males were maximizing their seminal fluid under high competition. So this data here is not as intuitive as the data on the previous slides, so I'm going to spend a bit of time taking you through it. So this is looking at the relative abundance of individual seminal fluid proteins Each vertical row here is an individual protein. The hotter the colors, the more there is. And these broadly form into two different clusters. There were a couple of additional clusters, but they were tiny and too complicated to speak about. And what you can see first of all is that these two clusters behave. Oh no, I'll come on to that a little bit later. So in cluster one, what we see is that there is greater production of seminal fluid in these virgin males that are exposed to high levels of competition. So that's where all the hottest colors up there. That's hotter than the low or no competition. And what we see in cluster two is evidence of greater depletion in the high competition treatment here. So there's sort of two different things going on here. For some proteins, males are just making more, but they're actually, after mating, it seems like there's no differences. So it suggests they're making more and then transferring that sort of excess of seminal fluid protein. And then in this cluster, they don't make any additional protein, but somehow they manage to transfer a larger quantity to females. So in either scenario, you've got females getting more protein when the males are exposed to high levels of competition relative to when males are exposed to no competition or low competition. So to try and bring together the sperm and the seminal fluid data into one sort of schematic, this was rejected by the reviewers as being rubbish, but I like it, so I put it in all my talks. This is a kind of a cartoon of the amount of stuff that males are transferring to females, but the sperm goes up under low competition and then maybe slightly down again. For seminal fluid, it doesn't change between no and low competition, it only goes up with high. So this was really cool. We're very excited about this, but we didn't know why males might be doing this sort of thing. And a bit of a spoiler, we don't still really know why males are doing these particular patterns. But we did get some sort of slight clues as to what's going on. There was a small but sort of marginally significant effect on the number of offspring produced by females. So if males have been exposed to high competition, then the females they mated with produce slightly more offspring in the days following mating. So this is what you might expect. They're transferring more stuff. Females produce more eggs. But then Ben Hopkins followed this up by mating those males serially to 5 females in a row. And by the time they get to the 5th female, then the patterns have changed somewhat. So it seems like the males who hadn't been exposed to rivals are then doing a bit better. And the males that have been exposed to rivals are doing worse. So it's kind of consistent with the idea that exposure to rivals, you put loads of effort into the first mating you get, and then subsequent matings you suffer because you haven't got so many ejaculate resources left. And this was kind of backed up by the finding that if we just look at the mating behavior, males were pretty good at mating despite whatever competition level they came from with the first female. By the time they got to the 5th female, the males from the high competition levels were kind of rubbish. They were slower to mate with females, and fewer of them even managed mating at all. So again, consistent with the idea that there's a high cost to putting extra resources into the first mating. So the take-home messages really from this story were that male Drosophila are sensitive to their perceived competition level, and in response to that, they can alter their ejaculate composition, and they have distinct rules for what they do with sperm and the seminal proteome that we haven't fully disentangled the reasons for as yet, but it seems to come with distinct costs in terms of not being able to mate with so many females, but benefits in terms of extra stimulation of fecundity with the first female. So this is what I've tried to show you here is some examples of some of the new insights we got into fly reproductive biology from quantitative proteomics. So we were able to identify some new seminal fluid proteins, and our approach has now been used by some other groups in different species to try and find new seminal proteins. The work I just talked about here is about, showed you how sperm and seminal fluid changed in response to rivals. We've also used it to try and understand stuff about cell secretory function. So we used some mutants where some of the seminal fluid producing cells didn't function properly, and tried to understand what happened to the seminal proteome. That was quite fun. And I've also used it to look what happens with male aging and the relative role of sperm and seminal fluid in reproductive decline. That was a nice lesson in your theory being completely wrong. So I wrote a grant about how seminal fluid depletion might explain male reproductive decline, and it doesn't at all, not even the slightest. But what we did find was that maybe seminal fluid proteins end up getting degraded and cleaved and showing post-translational modifications when they get old. And yeah, we've also looked at how these things called seminal microcarriers, basically seminal fluid is not like floating around in the accessory glands, it's actually packaged up into little bundles. And you can manipulate that with various mutants, and we looked to see how the seminal proteome changed when we messed around with that. And then we've got a couple of ongoing projects where I've been looking, I spoke today, somebody's looking at larval crowding, and we've been looking at how seminal fluid protein production and transfer changes in males that have been from crowded or uncrowded environments. And we're looking at how long-lived males' seminal fluid and sperm changes with time. And other groups have been doing this, these sorts of ideas, in humans. So trying to associate changes in the seminal fluid and sperm proteomes with male infertility. So maybe we'll get that answer to who's killing America's sperm. Maybe it will be the seminal fluid. I don't know. Anyway, that's all of my talk. The people who did most of the work that I'm talked about today were Ben Hopkins, Iram Seppel, and Ellie Barth, a whole bunch of undergraduate students and people. These are the people from the proteomics facility in Oxford. Thanks very much.